Hi viewers, my name is Uncle P and you are on to ISIFE EDUBLOG. In today's video, we are going to be analyzing or interpreting this year's SSCE WAEC Biology 3, that is practicals. In this video, we're going to be preempting to our viewers what they are expected to see in this forthcoming examination, which is scheduled to hold on next week, Tuesday. So we'll start with two specimens, and from this we'll advance to other specimens. The reason why we are looking at these two specimens side by side is due to their similarities as well as their differences. Okay, in this specimen A, where you have a mature fresh eggs of a catfish, and in specimen B, you have fresh egg of domestic fowl. Okay, we are going to be preempting what are likely going to be in the mind of these examiners. In this video, we shall unravel that. Okay, let's start with this question, and then you can pause the video and try and see if you can answer it. Okay, now, matured fresh eggs, remember eggs, so it means that the candidates will be given numerous eggs of specimen A, okay, and it's from catfish. You are not going to see any catfish around, okay, in the table, on the table where the specimens are displayed. What you see there is eggs of this catfish. So it does mean that the candidates are not to know where these eggs coming from. So it means that you're going to look at the egg and see if you can relate this egg to this organism. So it means that questions are going to be coming up to which of the organism lays, or this particular eggs, which type of vertebrates share or lay these eggs. And then that would be, you're not going to mention catfish. Maybe you see a question, mention the class of organism that lays these eggs, or mention the class of vertebrates that lays these eggs. Of course, you should know as a biology student that these eggs are laid not just by catfish. Catfish is one of the examples of organism that lays these eggs. But you say that this class of organism belong to the class we call Pisces. Okay, Pisces is one of the it's the first class of vertebrates. It then means that this particular organism that lay these eggs appears to be all aquatic. Yes, they are all aquatic. These are the kind of questions you're going to be seeing. So what will be in the mind of the examiner? The examiner wants to really know from you if you can relate these particular eggs to be that of fish, and specifically what type of fish. You may be asked to give an example of the class of vertebrate that actually lay this egg, and I can answer that it is the egg of catfish, supposing this question is asked in that manner. Okay, now having answered that question, you know that these particular organisms appear to be an aquatic organism, and then looking at how the eggs appears, some kind of observable feature. So two things you see some of the things you see that particular specimen, the eggs, how does it look like? Okay, I don't know whether you have seen a vitamin A tablet before. Okay, it's kind of a tablet, is yellow or golden yellow in color. That is exactly how these eggs of catfish look like. You may even mistaken it to be vitamin A tablets. You can get from a chemist shop. It is transparent yellow or golden yellow, very small, but it is in several masses. In fact, the particular organism lay this egg in masses, several masses of this egg, and it is transparent. It appears that you can look through the egg, okay, and the egg is without a shell. Because we're going to be comparing this egg with that of bird. Maybe in this video, you're going to see the egg of a fowl and then the egg of a matured catfish. And we are going to compare, look at the comparison, look at the similarities and the differences between specimen A and specimen B. I mean structural similarities and structural differences. And one of the things we're going to be looking at is compare the size of both eggs. One appears to be large, one appears to be small. Okay, you should pause the video and answer which of specimen A appears to be small size, which of specimen B appears to be big size, or which of specimen A has shell, or which of these do not have any shell. Okay, going forward in this video, I would also like to mention this is very important. I'm looking at where the examiner will want to know from the candidates or from the student if they can actually look at other observable feature of egg of a matured catfish. It does not have shell, as I mentioned. And then this egg appears to be laid in water, unfertilized. Okay, yes. Fish, not just catfish, generally fishes or any class of Pisces lays unfertilized egg. I mean eggs that are not fertilized. Okay, what this means that this particular organism lay this egg. They are not laying this egg on the ground, they are laying it in water, being that the absence of shell in these eggs of catfish could have possibly caused some of the liquid contents of this egg to dehydrate due to absence of shell. Okay, so it means that this particular organism must be well adapted in aquatic environments where these eggs are laid to avoid the eggs being dry or desiccate. Okay, they lay this egg in water, numerous eggs in water, knowing fully well that the mortality rates of organisms that belongs to this class of vertebrates is very high. So to compensate the mortality rates, this particular class of organism lays numerous eggs. And the eggs they are laying is not eggs that are fertilized internally, but eggs that will be fertilized outside the body of this particular specimen A. What are the interests the examiner is looking out for? They may ask you to state the type of fertilization in specimen A. So since they lay unfertilized egg, it does mean that the male counterpart, the male catfish, have to deposit some kind of quality sperm on this egg when they are laid. So it means that since sperm cells appears not to permeate into this organism, rather this particular sperm cell are poured on the egg that were laid. This class of vertebrates simply undergo a type of fertilization called external. So fertilization in specimen A is external. External fertilization. External fertilization. In external fertilization, unfertilized egg are laid by this class, not just catfish, bony fish. These are the class of organism that belongs to this category that lays unfertilized egg. Now, if you look at the class of vertebrate, that is animal with backbone, where this catfish also belongs, you see there are other class of vertebrate like amphibians. Amphibians somehow also do the same. They lay eggs that are not fertilized internally. So eggs are later fertilized out of the body of the female. That means the female lays unfertilized egg, which were later fertilized by the male external. So the type of fertilization in specimen A is external. You're going to earn 1 mark for us giving this answer vividly like this. Okay, now there are so many disadvantages to this external fertilization. Okay, so many disadvantages occur here. I'm also seeing the fact that they may ask you another question: can you give an example of other organisms that still undergo this type of fertilization apart from the aforementioned specimen? And then this is the point you cannot give other example of class, which is amphibian, example toad or frog. Also lays unfertilized eggs, so they also exhibit external fertilization. Supposing they ask you to give examples, so example was trying to explore your mind to know if apart from fish, is there any organism that also exhibits external fertilization? Yes, you can answer it and end your mark for that. Okay. Furthermore, I want to say this now. This has a lot of disadvantage. External fertilization is so disadvantageous to organism that does that. Because at the point of laying unfertilized egg, which is to be fertilized out of the body of the female catfish, it does means that so much sperm cell discharged or released by the male, some of them will be wasted. And upon the time these eggs are laid, it could have been consumed by some predators. Some animals can consume them. So you now find out that there are very few of these eggs that will be opportune to be fertilized by this sperm. So so many sperms will be wasted, and so many eggs will also not be able to make it. But good thing is that most organisms that exhibit this type of fertilization is a very rare case of fertilization, must lay numerous eggs in order to compensate for these losses, which must definitely happen. So this is the reason why this aspect of fertilization will not that affect the organism, in as much as sperms are wasted, and as well as some of these eggs are damaged, some of these eggs are eaten also by some other kind of animal. So in as much as it is disadvantage, so we be asked to give disadvantage, mention to disadvantage of this type of fertilization. One, you can just see that a lot of sperms will be wasted. Okay, and again, you can also see that some of these eggs are also eaten up by other animals. So not all will be fertilized when it comes in terms of fertilization. So this is the disadvantage of this type of fertilization. And again, to compensate on these disadvantages, the organism that exhibit this type must lay numerous eggs. Okay, that is on that for specimen A. But I'm still going to come back to specimen A when I'm trying to compare it with that of specimen B. Okay, coming to specimen B now, which is a fresh egg of a domestic fowl, raw one. Okay, just like in specimen A, in specimen B, that one is very common, I think, to this to our viewers, which are candidates. I believe you might have come across this one. I think we are familiar with this than this. Okay, I mean that we know our normal egg, say raw egg. So it means that it might be a cooked egg or just raw one. So looking at the egg, the oval shape of the egg with shell. Okay, looking at the two eggs, A and B. Comparing both by size, there's structural observable difference, which I'm still going to be listing under differences between specimen A and B in terms of the size. It appears that this has a much bigger size than specimen A. Okay, but looking at egg laid by fowl, you can ask, since they are mentioned fowl, they may ask me a simple question like this, which is, give example of class of organism that produces this egg. What are the class of organisms that produce this egg? The class of organisms that produce this egg are what? Apes. An example, you cannot mention fowl. Other examples apart from fowl, you can mention pigeon, you can mention duck. Okay, these are the class of organisms that belong to this class called what? Apes. Apes are also vertebrates, that is animal with backbone. If you look at the features of these apes, you also find that they have their mouth modified into a beak, and then their bodies are covered with feathers. Okay, try to look at the egg laid by these members here. You will find that this egg laid by apes are fertilized already. Okay, birds lays fertilized egg, not like specimen A of fish that lays unfertilized egg, whereby the fertilization is external fertilization. In specimen B, examiner may ask you to state the type of fertilization exhibited by specimen B. In specimen B, what they exhibited, the type is internal fertilization. Internal fertilization. Okay, that's what it is, internal fertilization. Of course, you know the opposite is external. In internal fertilization, that means the egg is not needed to be laid before it's fertilized. So the egg stays there mature, then before the maturation of the egg, that means the sperm cell, the sperm of the male ejaculated in and then cause fertilization to take place internally, and whereby after the development of the egg, these eggs are now laid, okay, fertilized. Okay, now the eggs are laid, fertilized. So in as much as they lay their own egg, it does not mean that the eggs laid are just infertile. The egg already has been fertilized. What is needed there is that this particular class of organism, basically fowl, need to now incubate this egg by themselves. Okay, and the incubation period will just last between 21 to 25 days, after which the fowl now hatches its young ones after this number of days, particularly 21 days. So the type of fertilization in specimen B is internal fertilization, while the type of fertilization in specimen A is external fertilization. You may give an example of other class of vertebrates, animal with backbone, that also exhibit internal fertilization apart from apes. They can also mention others are reptile. Example, we have something like lizard. Okay, have lizard. Then we also have another class of organisms that also exhibit this type, which is mammals. Mammals also exhibit this type. Example of mammals that exhibit this type, one can even mention rats, even man. So these are the question. Definitely, viewers, I want you to concentrate on this. These are going to be in the mind of these examiners. Tell you, this is how you're going to see it. So this is on this note. And then mind you, we're going to be looking at the diagram of these specimens, that is specimen A and specimen B. Then from the diagram, we're going to try to look at the similarities between A and B, as well as the differences between A and B. I mean structural similarities and structural differences. Alright, so you now have the diagram of specimen A and specimen B on the board. You can pause the video and probably look at the two specimens, identify which one is specimen A and which one is specimen B. Okay, as you can see, I'll start with magnification here because it's very important in biological diagram. Magnification, the title of the diagram. Every diagram is expected to have a title. okay, which somehow can be up down differently depending on what you choose to write up there. Okay, this is fresh egg or domestic fowl. That is again so that the examiner can see what is to mark or to judge you of. Okay, at the end there, what aspect of the egg do we actually put out there, and based on the what was exactly asked in the question paper, because it appears you're going to draw of any of the sections, either a transverse section or longitudinal section. So if you are drawing by this cross-sectional area like this, cutting from up axis down, this is how it will appear. So you have to state it down here. And this is longitudinal section of specimen B, which appears to be a fresh egg of domestic fowl. Okay, that is on the issue of title of the diagram. Okay, I have the fresh egg and I have the exact what you are drawing. So let's count this way. This one, this is a fresh egg. This is the aspect of the egg you are drawing. So there are other aspects which can be maybe considered by the examiner, depending on which of the question the examiner want to throw out there. So you also have this one coming at the left hand side. This is the egg of a matured egg. You now see that it also has a fresh egg of catfish, and the specimen A. Basically, may not think that this diagram of specimen A, they will ask. I think if they won't ask, they will ask to draw specimen B rather than specimen A. But we never know which of this diagram will be. They will ask. So that's why we have the two diagrams on the board. So you can pause the video and try to check out how the diagram is. This is how best to draw. And you must know in this video, we're going to tell you how to draw effectively to end mark. So each time you write biological exam, try and see question assigned to biology diagram. You see 10 marks on it. You may start to lose all the 10 marks if you're not imbibed by the rubrics of drawing biological diagram. You must not be a super artist to draw to end mark in biology. You may draw something that is very fantastic and you lose all the mark. You may draw something that is not good. Rather that particular diagram look exactly, the examiner will know from the diagram that this is actually a diagram of a fresh matured egg of fowl. Rather, if you draw something that looks like table tennis, you can't end mark of such. So it appears that this two diagram here appears to demonstrate what A and B exactly look like. This is that of a catfish. Okay, it's very small. I think it's measuring it's measuring at, it's not even up to a centimeter. That's why it will be difficult to draw this. So that's why here is that it does appear that I'm magnifying it to times 10 or times 20 of this is not that specific. This is not specific, but I'm assuming that I'm magnifying it 10 or 15 times of its original size. Okay. Why here you can do this, you can get your fresh raw egg and use your meter rule to take a size. No matter how big the egg will appear, it will not be as big to exceed 5 centimeter or 10 centimeter at most. I think egg that is of 10 centimeter, I will be scared of that kind of egg. But let's just be frank here. The size of egg, let's keep it at 5 centimeter upwards. So the size of the diagram of paper will always be draw 8 to 10 cm. Okay, 8 to 10 cm. So you are looking at diagram of paper over what you have there. Life. Okay, suppose you have life 5, 10, that means 10 divided by 5. So that's why what we have here on this board is times 2 or times 3 or times 4 of the original size of egg of a fowl. Okay, look at what you see here on it. It appears that this one does not have any shell. That is, bear in mind the examiner is going to ask to differentiate to states 2 or 4 structural differences between specimen A and B. One of the structural differences is that in this case, what acts as a wall here is chorion. Okay, but this chorion is not a shell, it's just like gel-like material. If you have not already have touched vitamin A before, if if you touch vitamin E, it appears that it's very soft. It's just like gel, and that is chorion. But it appears that the shell here, the outer coat of this egg, is shell. Okay, what is the role of this shell in this particular egg? The role of the shell is to protect the contents. There's contents here. It appears that this particular inside of this egg is liquid. Okay, having egg white and yolk. Okay, is liquid. That is when it's not cooked. Okay, so to this shell protects the content, the liquid content of this egg from drying up or from desiccating. That is the function, that is the adaptation of shell. Okay, because it appears that organism that actually produces this egg are terrestrial organism, which there will be a problem of how this egg will be protected. So this is what the role of shell, the purpose of the shell is to prevent the content, the liquid content of this egg from drying up or from desiccating. Okay, but in this case, this is laid by aquatic organism, which does not have problem of drying up. So this egg still stays in water, so there won't be need for having shell. What tends to protect the liquid content here is the chorion, a gel-like material. Okay, now these are the comparison. So you can say that this possession of shell and there's absence of shell. Rather, what is acting as a shell here is a gel-like material called the chorion. Here you have the oil globule. Okay, if you look at the egg of a fresh egg, you now see that it's like something that is shiny like oily. That oily globule is what gives this egg that transparency. It looks as if you are trying to look to see the yolk and the egg whites of this. The yolk, it appears that you are seeing the yolk yellow. In fact, the yellow, the color of the yolk is even seen from the outside, meaning that the oil globule make it possible for the chorion to excite this color. And that's how you see this color appears. And you can even use it for differentiating the both eggs. Okay, looking at this egg, you can look at egg and find what actually is the color. Some egg appears to be light brown. Some egg appears to be brown. Some egg appears to be faint brown. So these are the kind of color, but this color is transparent. You cannot see yolk in it, but it appears that you're seeing this yolk, since this oil group is exciting the chorion. Okay, and then it appears that here you have a germinal disc. A germinal disc. The germinal disc is in there in the yolk. Basically, it appears that this particular egg, for this to be there, that means this egg is fertilized. Okay, it has kind of matured, and it has reached to the fact that this particular germinal disc is just like a part of the yolk that furnishes or that supplies food to the embryo that eventually will be here. Because it does appear that the embryo stays here. The yolk acts as the yolk contains 6 classes of food. So it is in the yolk that the young one or the embryo here survive, or the zygote here survive before it reaches this. So these are the discs that allow the what do you call it? This yolk makes sure it nourishes, it furnishes the embryo or the zygote from food, since it contains 6 classes of food. Okay, you have the chalaza here, you have the chalaza, and then you have the egg space. There is no chalaza here, there is no egg space here, but both have vitelline membrane or yolk. Both have vitelline membrane or yolk. Okay, these are structural similarities between these two organisms. Okay, in terms of size, this has large size, this have small size. This have large size, this have small size. Then, in terms of color, the color partitions in the specimen B here, have that the parts of the egg white here, okay, this particular surface after the shell is thin egg albumin. Okay, this region where you have the colors here is also white, but kind of the white keep fading to assume the color of this yolk. So it is thick albumin. Okay, the function of the calaza here, the two calazas here, is to position the yolk, to hold the yolk in a steady position, and then there is an air space. Okay, air space in between, which also give buoyancy in the egg. So look at its similarities, structural similarities. Give two structural similarities between specimen A and specimen B. Please compare the sizes and the color. The sizes, small size, large size. Okay, make sure it's in tabular form. A and B, small size, B, large size. Another differences again is that the touch. Okay, it is jelly-like or soft, hard. Soft, hard, soft, hard. Very simple, as brief as possible. Okay, you can also see another difference in there. The soft part is chorion, the hard part is shell. Okay, another point you can see through here is transparent, non-transparent. So these are the structural differences between specimen A and specimen B. But if they ask you to just the difference between the two specimens, everything can go. This specimen is laid by a class of vertebrates, which is catfish, is laid by other class of vertebrates, which is fowl. Okay, so in other things here, okay, you can see from the labial parts, you can give up to 7 or 8 differences between the both. There's similarity between the two. You don't need table during when you are similarizing to object or specimen. Okay, you can look at it to say that both, in terms of structural, both possesses what? Yolk. Okay, both possess yolk. It does appear that this one already has an embryo that is already forming. This can also house an embryo through the germinal disc. Okay, and vitelline membrane. Okay, vitelline membrane is just like the membrane bond, the membrane that bond the yolk. You also see this can also be vitelline membrane. So both also possess a vitelline membrane. So these are the similarities between specimen A and specimen B. And now we'll go to specimen C. Specimen C. Okay, specimen C, specimen D. Specimen C is uterus containing a fetus. Specimen D is longitudinal section of ovary or pride of Barbados flower. Okay, we're going to be examining the two specimens and try to also find out what will likely be in the mind of these examiners. Let's start with specimen C. Okay, uterus containing a fetus. This can be improvised. That can be drawn in a cardboard paper or a model of this, having a fetus that has stayed there for 10 weeks or thereabouts in it. Once it has reached that 10 weeks period, it's no longer a zygote. It's now at that point called a fetus. Okay, so this is exactly what you will see. Okay, in the model structure, this is what it appears. You see. In the case where you have to draw specimen C, you put so many things into consideration. Remember what I've told you in the earlier diagram, is that it appears that you give a caption to what you want to draw. Okay, the caption will be uterus containing the fetus. And you can also write the same thing here. Make sure you add labels, caption and title of diagram. Any of this can come in. Remember your size, 8 to 10 cm will always be what they are asking. Okay, it appears that the size of this in a model, supposing it is provided, the size of this in a model is actually going to be much bigger than the size of what you should draw. So it appears that they're going to draw half of the size of this, so which means magnification of this. Supposing you're asked to draw specimen C will be equals to times 1 over 2. Okay, it's going to be times half because it appears that this is big. Okay, these are the things you see here. Okay, let's discuss some of the functions of this. This is called the womb or uterus. At this our level, we refer to as this as uterus. But at advanced level, maybe those of you that will end up studying medicine, this will be called womb at that point, at that level. This is a fallopian tube. This is what it is called at this point. Okay, at this point, at the kind of advanced level, it's called fallopian tube, but our level, it should be called oviducts. Okay, oviducts. So that means the contents here, there was a time it was here as egg. Okay, this is where egg appears. Okay, we call it, we don't call it egg in this level, we call it ova. So this is the ova, which eventually becomes this. Okay, so but when this ova got to react with a sperm cell, okay, it produces a zygote, which stays in the yolk of the egg of this specimen until it moves from this point to this point where it settles down. The point of trying to settle, the size of this, something that has been in that form of zygote, having taken enough nourishment or food from the yolk, the size of this will not be comfortable again to accommodate the zygote. So the zygote will forcefully leave this position and try to reside to another position. Okay, this is struggle. This is more, but at that stage, it has now changed to what we call embryo. Embryo is at that point. At that point of embryo, okay, part of the tissues of the embryo with that of the womb or uterus eventually forms this, which is called the placenta. The placenta. I'm just trying to tell you the what we call functions or adaptation of these larvae parts. I told you the function of the fallopian tube or oviducts is to hold mature eggs that are mature enough to be fertilized by a spermatozoa. That is the function of the fallopian tube. It's pair, okay, both sides. So the moment this egg is fertilized, at a point, the zygote will move down when it is up to an embryonic stage to the uterus, where it is attached to the womb by the placenta. Subsequently, it is through this placenta that this zygote, embryo, gets nourishment from the mother. Both nutrients, exchange of gases, exchange of nutrients, exchange of fertile phases, fertile wastes, and all that got exchanged through the mother, through the placenta. So it's a very, very important organ of this. It's an organ that is within this womb as well, which ensures that food, nutrients, and all that are exchanged between this fetus or embryo to the mother. It is that point it starts to change at 10 weeks to what we have as embryo, which is a miniature of the mother. Okay, the uterus is where this fetus reside until gestation period. But there are so many factors that are also encouraging this fetus to stay here, such as the amniotic sac, which secretes amniotic fluid. Okay, the function of the amniotic sac is to secrete amniotic fluid. Then the function of the amniotic fluid is to act as aquatic environments for the zygotes. For the fetus. The fetus must swim here, so it means all part of what you see are liquid medium. So you provide a liquid medium for the fetus to survive. The fetus must be in this kind of position for its survival. So in that case, why this particular amniotic sac is in fluid form, it It also ensures that it prevents this particular fetus from mechanical injury. And then we have the cervix, which is also called the neck of the vagina. The neck of the vagina. Most often at this stage, this cervix is not that open. It's closed at this point, because if it's assumed to be open, that means some microorganism can get access to this particular position if this particular portion is closed. So at that point, it is closed, making sure that microorganism does not affect it. But once the womb is getting enlarged, it is forcefully making this neck of vagina or cervix to open. That is close to level of gestation period reaching. Then why it is called neck of vagina? Because it is through here that this particular fetus, once gestation period has reached here, fuses its head here, and then due to an enlargement of this, and the bones of the pelvic will allow it to do during child level, and through the vagina this child comes out. So the role of the cervix is to open and help during gestation period. It's also the neck of the vagina. Already know the neck of the vagina. Also, the role of the vagina is to allow the intromittent organ to permeate during the actual ejaculation of spermatozoa. So it's also there through the that urine is also discharged to the outside or through the urethra. So these are basically okay. Let's try to go back to specimen A and B and see what we talked about. Relate specimen C, specimen A and specimen B. Looking at this particular organism. Now look at the question examiner is going to ask you. Okay, the organism that has this kind of structure now, can you give the class the organism with the structure, the class they belong? If this class of organism are basically vertebrate, animal with backbone, but what class do they belong? All mammals exhibit this type of reproduction. Okay, so this is particularly possible in mammals. So it is a mammal that you see the setup. So the class of organism that exhibits this type of reproduction, that is not when eggs are laid and hatched, and that is mammals. So can you give example of mammals? It is there, and I'll give some examples of small mammals like rabbit, like guinea pig, and or what have you. So this is mammals that exhibit this type. Then it appears that the eggs are developed into the stages that are being laid. So what type of fertilization is exhibited here? Since the eggs were fertilized inside and which develop into the stage, so the fertilization here is internal, just like in specimen B, which is also internal. But the only difference between that of fowl with that of specimen C is that in specimen C, which is what we have on the board here, fertilized eggs were not laid. Okay, so here they are giving birth to, not where eggs are laid. So that is the difference between this type of fertilization and that of birth. So this way we have oviparity, ovoviviparity, and then viviparity. So this is a clear case of viviparity. So this is a clear case of viviparous organism. So in this case of reproduction, eggs are not laid. Okay, eggs are not laid. This is internal, but they don't lay egg. But in the case of birds, eggs, that is fowl, it's specimen B, eggs are laid, fertilized, and then hatched into young ones. So there's a clear difference there. Eggs are laid. So when we want to describe that kind of internal fertilization, we use the word ovovivi. So that is for specimen B. Ovo for fowls, ovovivi. Just like this, just like the ovo here is the only difference between that of vivi and that of birds. Ovovivi means that they lay egg. They actually lay egg just like this specimen A lay egg, but the difference in the egg laying here is that egg here are laid, fertilized. But in the case of specimen A, which lays eggs that are not fertilized, just like sterile eggs they lay, okay, which eventually basically become fertilized short time. Okay, that one could use just ovi to describe those ones, oviparity. So oviparity is a type of fertilization which is also a form of external fertilization in specimen A. So these are the kind of comparison we are looking at between the two specimen, the three specimen now. Okay, mention the organism that exhibit this type. The first one is both are all vertebrates, animal with backbone. Okay, the other one is for fish. The other one is for birds, and this one now is for mammal. So we have looked at all the functions of this labeled part. And there's something I want to also explain in this video, which is very, very important. So if you look at these three organisms now, specimen A, B, C. Now, you're looking at the number of eggs they lay or they produce. In specimen A, they lay numerous eggs. They lay numerous eggs. And then at the end of laying numerous eggs, some very few were fertilized. And I've mentioned in this video the reason why this particular organism should lay that number of eggs is because of mortality rates in those kind of organisms is very high. So that's why they must lay that kind of number of eggs to compensate for those that will die. So that's why if you're going down from what do you call it, from fish to birds and then to mammals, mortality rates is now decreasing. So when you come to birds, they lay eggs, but the number of eggs are very few. Okay, birds are specimen B lay few eggs, and the specimen C now do not lay egg. Okay, even compared to the number of organisms that this produce, you can't compare it with that of birds. So birds produce more number of organisms than mammals, than any class of mammal. So you now see that there is a reduction in the number of offspring produced by this class of organisms. In case if you're asked to arrange this in order of increasing or decreasing number of organisms produced by specimen A, B, and what? And C. And then in terms of evolution as well, which in evolvement, since all these organisms A, B, C, and D, they all belong to the same class, class of vertebrates. It's plain in evolution which one first evolved. The first one to evolve is catfish. Okay, catfish evolved first. It evolved for that specimen A evolved first, and then it is followed by specimen D before specimen C. That is how they evolve. Specimen C appears to be the last vertebrate on Earth. Specimen C, the last vertebrate on Earth. That's where you have mammal. Then I can see questions like parental care. Okay, parental care. Can you explain the nature of parental care in specimen A, B, C? There is no parental care functioning or existing in specimen A. There is no parental care. In B, yes, there is parental care, but it's not as extensive as that of specimen C. Because specimen C appears to take care of the young ones. Specimen B also take care of the young one, but it's not as extensive as that of specimen C. Please take note of all these points. These are silent points that the examiners will want to exploit vulnerable candidates. Okay, watch this, comment, watch this, and make sure you internalize all this analysis before the exam. Okay, with that, let's quickly look at the other specimen, specimen D. Okay, specimen D is totally quite different from this specimen, so I don't think we will have any basis of comparison. But if there's any comparison, we'll also mention that. So this is specimen C. Specimen D. Make sure these diagrams you learn all, because you don't know the actual diagram. They have a mind of bringing out. Okay, you should learn all the specimen diagrams so you can be in tone with the diagram. So this is specimen D, and it is a longitudinal section of ovary of pride of Barbados or flamboyant flower. This is the diagram. Okay, now this is a flamboyant flower cut at this axis. Okay, let's see what observable features. Okay, observable features. What can you observe? Let's come to the color. You will see the flower that they are thinking. This is rainy season, so you see this flower in abundance. I don't think your biology teacher will not make improvisation of this. It's going to provide it because this now is in abundance. So you can see what is the color? It is red. Okay, it's red or pink color. Okay, the color is an adaptation to invite insects for pollination. This is what you observe structurally. And they also observe, try to count how many number of anthers. Anthers, I think 9 or 10. Anthers, but can just show these two anthers, very important anthers and filament. These two anther and filament. The anther and filament is the male reproductive structure or organ of this flower. They will ask you to mention two male reproductive organs in this flower. Because the answer is anther and filament. Two male reproductive organ in this flower is anther and then filament. Okay, it's important you know that. Just like we have male reproductive, the two female reproductive organs of this flower are the stigma, look at them here, the stigma and then the star. The stigma and the star. This is the two female, this is the two male. Now look at how close this both appears. Okay, let's look at the function of this reproductive organ because this flower reproduces asexually. So you now look at this flower, but there are some other parts of the flower that are not essential, but they also play a very vital role in this flower. This is this two you are seeing here. That is the sepal and the petal. You must also observe the color. Okay, you appear that you see these particular parts of this plant green. This is in green in color. So the green nature of this means that there are numerous chlorophyll here. So this is sites of photosynthesis. So photosynthesis occur here. This is particularly the part that is brightly colored. That is the petal. Okay, that is the petal. So these two parts now is forming the non-essential part because they are not reproductive, but they play a vital role in the reproduction of this flower because this particular part can come together and become a petaloid. Okay, inviting insects due to their color and the kind of scent they produce. So insects are invited, and insects now on invitation causes pollination to occur without even knowing that it's going on. So in this case, in a flower, an insect may come for a particular substance. What is the substance that will attract insects to this flower? It is nectar. The sugary substance is nectar. So this is what invites this insect. This insect that pollinates this flower knowingly, they may ask you to mention the couple of insects that will always visit this flower for pollination. You can mention two insects like butterfly. You can also mention bee. Okay, so these two insects I just mentioned now must have adaptation in order to exploit this nectar. And one of the adaptation is they must have a long mouth or long proboscis, okay, which can pierce and get to the position of the nectar. Okay, nectar is a sticky, sugary substance, okay, that's when they get it, they feed on it, and then some of these nectaries stick on the body of this insect, butterfly or bee, so they can fly to other location of this flower. They can leave this particular part they perch, okay, and then go to this particular part, okay, and then this particular they now on doing that stick some pollen grain now stick, okay, on going there, the pollen grain now drop here and now diffuse through here, okay, diffusing through here, they start through the stigma. So that's why this process of taking one particular pollen grain, which is the male gametes, male sex gametes, okay, picking it from here to here causes what we refer to as pollination. So this is where I have now pollination. Okay, this is now when you should know what is pollination. Okay, the transfer of matured pollen grain from the anther of the flower to the stigma of a flower of a particular species. Though we can have two types of pollination. Now, what has happened here now, from here to here, appears to be the same flower. So this is called self-pollination. But in this kind of, there could also be possibility that cross-pollination can also be possible here. Yes, cross-pollination can be possible. What if the insect decided not to drop here and then fly to another of this flower of the same species, maybe this one now, and also pollinate it? So it means that this is coming from anther of different flower to the stigma of another flower, but two are of the same species. One problem with a cross-pollination is that what if on leaving this particular part to another flower, mistakenly drop the pollen grain to another flower that is not the same species with this? That means that pollen grain that was picked from here eventually is a waste, because it can't take a pollen grain from a particular flower, a sex garment from another flower, and go to another flower. Another flower will not recognize that particular thing, so there won't be any fertilization. There won't be any fertilization or pollination as the case may be, so it becomes waste. So one of the disadvantages of cross-pollination is that it wastes a lot of pollen grain. But here there is a sure banker that this will happen because it is the same. It goes here, so there is no mistake that happens here. So this is one difference between cross-pollination and then self-pollination. Then examiner want to exploit you on the type of ovary. Yeah. Okay, it doesn't appear. Look at the ovary, look at the ovule. Ovule is just like the seed. This is ovary. It appears what type of ovary does occur in this specimen. Now, because the ovary is above the sapele. Since it is above, look at the sapele, look at the sapele. Since the ovary is above the sapele, this ovary is superior. We have two types of ovary, can be inferior if it is below the sapele. But now that this ovary is above the sapele, this ovary is superior. So measure the type of ovary in specimen D. The type of ovary in specimen D is superior ovary. State the reason why it is superior ovary, because it is placed above the receptacle. It is also placed above the sapele. It is. Okay, mention one. Okay, it is an insect pollination. So mention the examples of insects that pollinate this flower. You gave something like butterfly, and then bees, and some other insect that will always come for nectar. And then the powerful adaptation of this insect that are coming is that they will have a very long mouth to get to the point of this nectar, which they use as food. The relationship between this flower and that of insects is a simple case of mutualism unseen by autism. Supposing such kind of question comes out, to look at the relationship that is existing between this and the insect. So it is a clear case of symbiotism. Symbiotism is an association between two organisms that are not related, but there is a mutual benefit that is going on between the two organisms. So a mutual benefit is going on between the flower and an insect. Both are not the same species, but they come, two of them benefiting in the sense that the insect benefits from the food and then causes pollination to occur. There are some kind of advantage that is given to flower that must be pollinated by insects. Such kind of advantages, they will ask, okay, you give reasons why this flower is pollinated by insects. It's because of the color. Any plant, any flower that is very dull, the insect will just come and pass by the flower. But any flower that is very, very colorful, so because of the bright color or brightness of this flower, insects are attracted, and then on doing so, it causes pollination to occur. So this is on this, and then please pay more attention on the diagram and make sure to learn this. Pause the video and look at how it is done, and make sure it is sketched with HB pencil to score. Okay, on the magnification of this flower, it appears that the magnification will be times 2 of what you are seeing, because I'm assuming that the size of this flower shouldn't go beyond 5. So looking at it, so you're going to draw double of it so that it will make sense. So to be double of it, which is mg, is times 2 of what the original size is. Okay, let's quickly look at this with that of specimen E in relation to flamboyant, because it's also in prior Barbados with that, but this time it is just the leaf of Barbados. Let me see.